Well, hi, everyone. Welcome to our presentation today on Callicarpa and various other topics. My name is Pam Soltis. I'm from the Florida Museum of Natural History at the University of Florida, and I'm going to be presenting, along with our team members here, some information about some of our research that involves Callicarpa, the beautyberry. So before we get started any further, I'd like to introduce the rest of our team. So we have Alex Abair, who's a graduate student in the Florida Museum in the Department of Biology. We have Doug Soltis, who is a curator in the Florida Museum of Natural History and a professor in the Department of Biology. We have Allie Bordini, who is an expert on public outreach, and she's been working with us for several years now on various outreach topics involving mints. And we have Grant Godden, who is a postdoc who's been working on this mint project with us for several years. He actually got his PhD at UF in our lab as well, working on mints at that point. And his master's degree was on mints, so he's quite the mint expert. So what we'd like to do today is share some of our information, some of the work that we've been doing on mint diversity and evolution, and we'll be focusing on Callicarpa. So at this point, I'd like to provide you with a little bit of background and an overview of our presentation. And then I'll pass things off to the rest of the team members. So we're going to focus on Callicarpa, but we're using it really as a way to describe some of the work that we're doing using genomic methods to learn more about mints. So one of the first things that our presentation will do is give you an introduction to mint diversity and evolution. And this is something that Alex will share with you shortly. But the mints are an incredibly diverse family, and there's a lot of mint diversity in Florida as well. And many of you, I'm sure, are familiar with some of our iconic mint genera, things like Dicerandra and Conradina that have federally and state listed species that have a number of species of high conservation concern, and many other of our amazing genera and species in Florida. But again, we will eventually focus in on Callicarpa. After an introduction to diversity by Alex and an introduction to genomics and its uses in mints more generally by Grant, Doug will provide a little bit of background on Callicarpa and then focus in on our genomic work on Callicarpa. So genomics offer a powerful tool for investigating many aspects in modern biology. And we can ask questions about how species are related to each other, and we can generate a phylogeny or an inference of the evolutionary history of a group of organisms. Now, the genomic methods can be used to provide us with data that we use to generate these phylogenies, but the genomic data can also be used to help us address questions about the evolutionary mechanisms that have generated diversity. One of these mechanisms that's very common in plants in particular, and also within the mints, is something called polyploidy, or whole genome duplication. And Doug will talk more about that a little bit later. Now, one of the most amazing things about mints is their chemistry. And this chemistry is, as we understand it, the basis of defense systems against herbivory by other organisms. But this chemistry can also provide the mints with new adaptations that then allow for new species to originate and for diversification to occur. So when we're focusing on the mints, one of our main points of emphasis is on their chemistry. So the project that we are part of is a larger collaboration that includes those of us at the University of Florida. There are also a few other graduate students and undergraduates who have also contributed to our increased understanding of mints, partially in Florida, but also more broadly. We also have collaborators at Michigan State University, Purdue University, and the Max Planck Institute for Chemical Ecology in Germany. So the parts that our lab is focusing on really have to do with the evolutionary aspects, and the other labs are providing the context of genome analysis and also chemistry. So with regard to our Mint Genome project, the first main aspect of this work was an initial analysis of the phylogeny of mint species. Now, as you'll hear from Alex in a little bit, the mint species is very large. And so when we constructed this first phylogenetic tree or family tree for the mints, we focused just on a subset of species, and we used information that came from expressed genes across the genome. And we focused on 48 species of mints. And again, this overall view of the evolutionary relationships within the mints is key to helping us understand how new chemistry has originated. Now, the mints, in fact, are a very large industry, over $200 million per year in terms of their agricultural and industrial value just within the US. Now, as a follow-up to that first project, we've been, for the last few years, constructing complete genomes of about 14 additional mint species. And some of these are listed here. We are including in our analyses peppermint and spearmint, some culinary herbs, ornamentals, and important genera such as lavender. Wood, teak, for example, is a member of the mint family. Chia is also in the mint family. And then, as you'll hear more from Grant, there's been some focus on some of the very important chemistry that is included in things like catnip. And also there are some herbals that may be important for medicinal reasons. And then, of course, there's Callicarpa. And as I mentioned, Doug will talk more about that in a little bit. So this is essentially an overview of our presentation following this very brief introduction, and I'm just about finished. I'll be passing things off to Alex, who will introduce mint diversity on a global scale. Then Grant will talk about the Mint Genome Project itself and some of the ways that genomics can help us understand specific questions. Then he'll pass things on to Doug, who will talk more about the Callicarpa project in particular. And then we'll wrap up with Allie providing an overview of how to make Beautyberry jelly and poptarts. And she'll direct you to some amazing videos that she's been developing that give snippets of information about the diversity of mints. So at this point, I'll turn things over to Alex, and he can introduce the extent of mint diversity.
Great, thank you, Pam. I'll switch this over. Okay, so yeah, I'll be talking about mint diversity today. As Pam mentioned, it's a huge family with over 7,000 species and 236 genera. The root word for the name is Lamiaceae. That means lip. That's in reference to the two lips on the corolla. You can see on the photo to the right here, there's an upper lip and a lower lip. Most mints have that character. Not all mints have that character, but it's the root of the original name. But yeah, it's the 6th largest flowering plant family, and that's pretty large. And you can see on this map that I got from GBIF, these are all occurrences of Lamiaceae across the globe. It has a very wide range, all sorts of habitats, not in the frozen tundra, not in Antarctica, but pretty much everywhere else. And there are a lot of rapid radiations where new species are proliferating in pockets across the globe. And if you look at your spice rack, you'll be surprised. Maybe you're not surprised, but there's a chance you'll be surprised at how many of these are actually mints. So everything from basil, thyme, lavender, all the way through marjoram, those are all mints. And a lot of products you encounter on a daily basis are mint plants. And there are other types of mints. So you've got plectranthus or coleus, as it's more commonly known, and you've got chia pets, some of these absurd ones, and then cat inebriants. You've probably, if you have a cat, you've probably given your cat catnip. Not all cats are susceptible to it, but a large proportion of them are. So I'll cover some of the general mint morphology before I go into some Florida mint examples. But on the right here, you've got a typical mint corolla. This is actually a salvia. This is chia itself, where there's an upper lip and a lower lip, and there's these lobes associated with it. But there's a common leaf morphology that you'll find, the opposite, alternately opposite, which is called decusate. And often, but not always, they have a square stem. Sometimes you'll find things in Asteraceae or Plantagenaceae that have a square stem that can kind of trick you. And they also have a variety of leaf forms. So sometimes they're simple, sometimes they're pinnately compound, sometimes they're palmately compound. And on the right here, you've got an example where there's a mint without a square stem. And then inflorescence types, there are a few common themes. There's terminal axillary, terminal capitate, which means head-like, and then axillary, which is generally the more common form. And fruit type is pretty universal. There are some exceptions, but usually it's these four-seeded nutlets. They're usually dry fruits that split into four pieces. But there are these oddballs. So on the left here, this is Philostegia from Hawaii that has a dark fleshy fruit at the bottom here. And then the middle, the star of the show today, which is Callicarpa americana. And then on the right here, you might have seen this in parts of Florida. This is called Clerodendrum indicum. And it has these blue fleshy fruits surrounded by the red calyx. So now I'll cover some of the, not all, but some of the genera that you'll find in Florida. There's a huge amount in Florida, and many of them are threatened, actually. So a lot of the Dicerangela, a lot of the Conradina are threatened in Florida. But there are over 150 taxa. And first, you've got scrub mints. These are very aromatic, very minty smell, kind of pennyroyal smell, if you're familiar with that. But they have these really ornate corollas with the maculation or spotting. And the corolla is kind of bent at a right angle here. That's one of the characters you look for. And then a monotypic, a single species in this genus, Stachydioma. This is another kind of slightly minty smelling mint. Trichostema, you might have seen this around town. There's a few species here. I think a couple are endemic to Florida, but they have these long curled filaments, which is the reference for blue curl. And then Pycnanthum, we've got a couple species here in Florida. Well, three, I think. There's a third one up in the Panhandle, but these are very minty, strong, pungent smell when you crush the leaves. We've got a few sages. This one on the top left is azure sage. It's giant blue sage, I think is the common name, but that's flowering right now. It's at the tail end right now. And water horehounds. These are commonly found at riversides. They don't have any sort of scent. They're just odorless, but clearly mints. They have these axillary clusters and very decusate leaf arrangement. And skullcaps are kind of an interesting group. They're unrelated to the other groups that I mentioned so far, but they're identified by these little protrusions on the calyx, and that's a reference to European skullcaps. I don't quite get the reference. I've looked at skullcaps. I don't really see the resemblance, but yeah, that's the derivation of that name. Oops. And Hyptis, this is the most common genus you'll see in Florida outside of Beautyberry. This one is at roadside. Well, both of these are at roadsides all over the place. On the left, this one has kind of a pleasant smell, but sometimes unpleasant. And the one on the right here is called musk mint. It has a slight musky smell. But these you'll find on the roadside all over the place. And I'll just end here with American Beautyberry. This is the one we're talking about today. You've got the flowers on the left here, which are atypical from most of the other mints because they're actinomorphic, meaning radially symmetrical rather than bilaterally symmetrical. And the stems are circular and the fruits are fleshy. So they're just kind of odd altogether. And when you look at the whole tree of mints, these are kind of distant from the rest of them. They're sort of sister to the rest of the mints. But with that, I will turn it over to Grant.
Thanks, Alex, for that really great overview of mint diversity. So I'm going to pick up and I'm going to talk a little bit about what we can learn from genomes and briefly describe some of the results that we've had that we've learned from these sequences in our exploration of these genomes. As you can see here from our selection of published papers that have come out of this project, we've learned a great deal about genomes and mints in general. And many new studies will come from these genome sequences. They serve as a resource for many other projects that will come after this project is finished. But because I only have 7 minutes to speak to you today, I can only share a small piece of the story. And so I wanted to first start with some background information about what we do in genomics. So most of what we learn in genomics, as Pam mentioned briefly earlier, comes from comparisons of different species and the sequences and attributes of those species. As I like to say, one genome is great, but many genomes are better. By choosing plants with different or contrasting attributes or traits, including chemistry, morphology, physiologies, or even yields or disease resistance in the case of crops, we can begin to tease apart the genes and genetic architecture underlying those traits. For example, we can make comparisons of gene content, including the presence and absence of individual genes. We can identify gene copy number and variations of those copy numbers, and expansions of individual gene families through duplication processes, either by tandem duplications where they occur next to each other on the genome, or via whole genome duplications, which Pam also mentioned. And in addition to that, we can identify lineage-specific genes that are unique to certain plant species or plant groups. We can also examine the structure of genomes, which I'm showing in these three circles here, including the distribution and order of genes, the presence of genes in gene clusters, which are often important in biosynthetic pathways or metabolism. And we can identify conserved or shared chromosomal regions that reflect ancestry in plants, including in different plant groups, species, or varieties of plants. We can also examine the regulation of genes and their expression in plants, which can help us identify candidate genes for traits of interest that we're focusing on, including chemistry in this case. But ultimately, our goals are to understand in an evolutionary framework the genetic basis for plant traits, and to understand how plant genomes change over time to yield the incredible diversity that we see in the plant world. And as Pam mentioned, we've sequenced a number of plant genomes so far. Here with red boxes, I'm indicating some of the ones that we've already published, including teak, which is a common timber tree, beautyberry, the catmints and their relatives, and culinary herbs. We also have a few other genomes that are currently in progress, including chia, where we focus on oil-producing qualities of those seeds, which are considered a superfood, and lavender, which is a very important plant in the fragrance industry. And interestingly, I want to point out this interesting fact that this is really only a drop in the bucket in terms of the number of genomes that exist. There are always some in the pipeline, but in terms of how many flowering plants we have in this world, it's a very small number. Only about 236 had been published just as a few years ago. So to walk you through that process that I just mentioned earlier on comparative genomics, I wanted to share with you a brief story, a mystery story called The Curious Case of Catmints. Many of you are probably familiar with catmints. There are multiple species of them. The most famous example of that is catnip itself, which we know contains a special class of volatile compounds called iridoids, or specifically naphthalactone, which I'll tell you more about in a second. This is, can affect the behavior of approximately 2/3 of cats, including many wild species such as tigers and lions and ocelots. And it induces behaviors such as cheek rubbing, rolling on the floor, and generally your cat will act pretty crazy when it comes in contact with catnip. But in nature, these compounds actually have an important role in protecting plants against insects that might eat them. So they have an important biological function as well as a fun cat behavioral inducing function. So we've known for some time, since some of the early mint studies that have attempted to reconstruct family-wide relationships, we've known that iridoid compounds are present in a lot of the species in about half the family, but in the largest group of mints, which we call the Nepetoideae, which include many common species that you know, like the culinary herbs, for example, as well as cat mints, iridoids were lost. So when we surveyed in the initial phase of this project, we did not find iridoid compounds in the majority of, well, in all of the species in this clade, except for in the genus Nepeta. And that's really interesting because it suggests that in the evolutionary history of this family, iridoids were lost and then remained lost throughout most of the family, but reemerged in the genus Nepeta, and possibly some other close relatives, but we haven't confirmed that yet. And so another interesting fact about this is that the biochemists that we work with have identified the different compounds and their abundance in these different, and we know that the iridoids that are present in this blue portion of the family are actually different type than we find in iridoids, the Nepeta-lactones, which have the cat behavior inducing properties. So why is that, and what did we do to figure this out? So we actually selected 3 species for genome sequencing. 2 of them are in the genus Nepeta, the catnips and the catmint. And the species Hyssopus officinalis, or hyssop, which doesn't have or doesn't produce iridoids. And this provides a contrasting case so we can look at the genetics and see what's different about plants. So from these 3 sequences, the first thing was to compare these sequences against another plant with the sequenced genome called Catharanthus roseus, or the periwinkle. And this actually has an established pathway worked out for irid production. So using what we know about those genes, we can compare with our genomes and by similarity come up with a list of candidate genes that might be involved in iridoid biosynthesis. And also by layering levels of information from the expressed portion of the genome. I don't know why we're moving forward. I cannot move backwards. I'm sorry, I've lost my connection here. So let me go back to this. So by layering information about gene expression using those groups of candidate genes, we could actually see which genes were overexpressed and then better identify the actual genes that are responsible for enzymatic activity. And then from there, our biochemists were able to validate in vitro experiments whether or not those individual enzymes produce the next product in the pathway. So doing this, we were able to use the information about Catharanthus to identify the genes and to validate those genes and their activity. The interesting thing is that we were also able to use analyses of genealogies, of enzyme activity, and of genome structure to reconstruct the chronology of events that led to emergence of iridoid biosynthesis in Nepeta. And one of the first things that we did was to look at gene genealogy that included iridoid synthase and related genes. And what this figure really shows, if you're not familiar with reading phylogenies, there's really one thing to take from this, is that the majority of mints who produce iridoids have iridoid synthase genes that cluster together in this single clade. And then those are related to, I don't know why this keeps happening, to another gene family involved in, can everyone still see this okay? I'm just gonna leave it like this because it keeps turning off. Are related to another clade of primary metabolism genes called the progesterone 5 beta reductase genes. And within that, a gene duplication event occurred, actually more than one. So you can see the gene for hyssop is here. Hyssopus, as I mentioned earlier, does not produce iridoids, but Nepeta does. We had a gene duplication that occurred right here, and we had diversification within that via more gene duplication events. So by dating those genes using molecular dating methods, we were able to estimate this event occurred approximately 23 million years ago. And after those genes evolved in iridoid synthase function, the genes were then translocated by a subsequent diversification event or duplication event that translocated that gene to a new place in the genome. And the old copy of this gene became redundant and was subsequently mutated until it became a pseudogene. So it's not functional anymore. And this provided evidence that indeed this did split from and gain a new function following duplication. And this is a common theme that we actually see in plants with unique chemistry. We have a duplication event followed by neofunctionalization is the term we use to describe the evolution of a new function. And then from there, new chemicals can evolve. So this concludes the story of how catnip new function. And I think the next step is really to understand within Nepeta itself, there are a number of different stereoisomers, and they have different properties. And the concentration and abundance of those different stereoisomers have different insect repelling properties. So that is to understand how different Nepeta species are producing these different stereoisomers, and that's a topic of ongoing investigation. So I think I provide a review of the general process that we and the genetic architecture of traits such as chemistry. And now Doug is going to tell you a little bit about our project with Callicarpa.
Thanks, Grant. Okay. I'm going to share my screen now, and we will continue with a closer look at the star of today's show, Beautyberry, which you've already seen. You've seen several photographs of this already. It's a shrub. Here's its distribution in the southeastern United States. But realize this is just one of about 165 species in this genus Callicarpa. It's really prominent in Southeastern Asia, but there are other species in Central America, the Caribbean, and they are remarkable plants, and we are interested in them for a number of reasons. So here's sort of a summary of the family tree of mints. It's a simplified tree, because keep in mind there's thousands of mint species. But if you look at this tree, here's some close relatives of the mints and everything up here. These are all mints, but Callicarpa is in this lineage down here, this group right here. So this is one of the early branching mints. So we wanted to know more about it. It's like, hey, it's our best chance to look at the earliest evolution of mints. And what this is going to be a story of is the importance of something called polyploidy or genome doubling. And that's shown or simplified here. So what you do is you have two initial species, one with orange chromosomes and one in blue. You can tell we're from the University of Florida here, orange, blue. Anyway, they hybridize, and then you get this diploid hybrid. Now that hybrid would be sterile on its own, but if you double it, if its chromosome number doubles, if it messes up in mitosis or meiosis, all of a sudden you have this thing at the bottom, which is a tetraploid, and that's a functioning new species. And so this has actually happened a lot in evolution. It's a very important phenomenon. And we'll show you here on the next slide just how important it is. So you are an ancient polyploid. vertebrates are the result of 2 old episodes of genome duplication. Flowering plants have 1 ancient duplication that they all share. So these are major events. They're super important. Next time you have a beer, realize that yeast was anciently duplicated. A lot of fish lineages have it as a very important evolutionary force, including salmonids. And virtually all of our crops are fairly recent polyploids. By that, I mean 10,000, 100,000 to several million years ago. And there's a selection of them there. So this is really, really important. And it turns out to be super important in the evolution of mints. So let's do some genomics. How would you tell? How would you know? So what we're doing is looking at a part of a genome, a small part of a genome. Realize that genomes are huge. You got thousands of genes that you're looking at. And if every gene is duplicated following polyploidy, you can pick up that signature by looking at the genome itself. And that's what we're doing here. We're looking at go back. You're looking at a part of the genome, 2 different chromosomes in the same genome. You can imagine this is Callicarpa. And here, all these red lines connect where the same gene is present multiple times. And not only are the genes present multiple times, they're also present in the same order. That's called synteny. So you have all these blocks of duplicated genes that are conserved. That is a cool, super clear indication that genome duplication has occurred. And so that's what you find when you look at the genome of Callicarpa. So Grant stressed the importance of comparing genomes. And so we went, wow, how about teak? Let's look at the genome of teak, which you can see also is down near the base of this family tree of mints. And we've looked at some of the other mints that have been mentioned by Alex and others during this talk. And if you look at the genome of teak, let's just compare that to the genome of Callicarpa. You get this thing that looks like a yarn disaster. You know, you can imagine all these different balls of yarn and what a mess. But each one of these is a chromosome. We have the chromosomes of Callicarpa on the top and the chromosomes of teak down at the bottom. And you can see just how conserved these are across these chromosomes. These are genes that are found in the corresponding chromosome of teak in the same gene order. So that genome duplication is present in teak. I'm trying to show you data here, right? So here's the Callicarpa genome here and the teak genome here, and you're matching them up, and you get these things that look like little worms, but those are actually conserved gene chromosome segments with genes. And you can see I put these little boxes on them that you can see that a lot of times you're getting worms in pairs. That means there's 2 copies in Callicarpa or Beautyberry, or 2 copies in teak. So it's really clear evidence that they share the same genome-wide duplication. So a little bit of genomics, what we actually do, how we use the data. So Grant also talked about the chemistry of mints. And so what we've learned in Callicarpa is that genome doubling event contributed to the expansion of a gene family. These are called terpene synthetase genes. They make terpenoids. And so the insect repellent activity of Callicarpa is due to the production of a number of these terpenoids, including a cool one that I like, Callicarpanol, actually named for the genus. And our chemists have actually helped identify the key genes that are identified to make these terpenoids. And so this polyploidy or genome doubling event was key in expanding those genes, expanding the chemical arsenal, if you will, of Callicarpa. So the take-home message, genome doubling rocks. This is a major evolutionary force. It made the mints what they are today. Hopefully you realize, hey, I can do genomics. I can look at a few things and I can understand how genomics work. These genome projects are really important because it allows the rapid discovery of genes that are encoding these plant-derived, really cool, important agrochemicals. And these genomes, as Grant mentioned, are really super important for understanding the chemical diversity in Lamiaceae in the mint family. So there's a lot of economic importance here. So I'll just end with this slide. The genome doubling is a key part of the mint story. And I will pass it on now. Allie, she's going to take us home with some really cool uses of Beautyberry.
All righty. So I'm Allie, or Allison, either is fine, and I am the outreach person for the Mint Genome Project. And one of my favorite things about the mints is that they have had so many historical uses for thousands of years. So this chemistry and this diversity lends itself to all these amazing uses all over the world, from India using tulsi, which is a species of basil in a lot of sacred traditions, or many of the mints like rosemary and oregano were used in mummification in ancient Egypt, or like lavender scenting bathhouses in ancient Rome. So we have all these uses using all this diversity in chemistry. And Beautyberry, Callicarpa americana specifically, is really cool because a lot of indigenous civilizations all over the Southeast United States used Callicarpa americana for medicinal purposes. And I really like getting into how people in the past used these awesome plants. So I'm not, making any medicinal potions or concoctions, or they would also use the leaves as a mosquito repellent, which I guess I could do. But I'm a huge sucker for baking for friends and getting things to as gifts, giving gifts, especially in the fall. So I made Beautyberry jelly, and I'd never made jelly before, but I'm going to go through just basically what I did. So I got 4 cups of washed berries and I boiled them. And this gets all those purple colors, the anthocyanins out, as well as many of those chemical compounds that we see in Callicarpa. Then, of course, you have to add sugar, and a lot of it. So I took about 3 cups of my juice and added 3 cups of sugar and dissolved it. And then we get to the fun part of adding lemon juice and pectin. And when you have sugar, which is polar, it will attract the water molecules. The lemon juice makes an acidic environment, meaning the pectin can bind together and make a jelly. So after simmering all that together, you get a very thick jelly. And anyone can do this at home. I personally followed my pectin's directions for blueberries, unrelated to Callicarpa americana, but it worked perfectly fine. And then you pour it into jars and give it to all your friends and family, and it tastes amazing, almost like a very sweet strawberry or raspberry jelly. But you're using a really cool plant to make your jelly. It's delicious. You have the beautiful purple colors from the beautyberry, and yes, you can eat that on bread or something, but I ended up making poptarts. So I made my own pastry flour and added cornstarch to my jelly, and it made it a nice thick Pop-Tart filling. And honestly, it's one of the best things I've ever created in my life. And I bake a lot. So I have these easy kind of step-by-step processes on a little series I've created during the pandemic called Mint Minutes. So I've made videos throughout these last few months on many members of the mint family, talking about diversity or some of the rare mints like Dicerandra. And so I do have videos. I'll have Valerie link them. But if you guys are interested in more of the videos besides the Callicarpa ones, they're all over on my Twitter, which is Allie Kokosai. I've pinned at the top all of my mint minutes in order. And, there's making cheddar thyme cornbread and, lavender Play-Doh, so many different things that you can do with the mints. And so I also have a link to my YouTube channel, which has a more in-depth look at beautyberry jelly. But anyway, I just love cooking or creating with the mints, and they're just such an amazing family. So that's that. So yeah, thank you so much. And wait, where's my cursor? There we go.
So thanks everyone for all of your contributions to, I'll put Doug back in there, for all of your contributions to our presentation. And thank you so much to everyone for joining us for the last 40 minutes. At this point, we are available to answer questions, and we will be happy to do so for the next bit of time.
Great. We have 2 questions in the chat now. So Diane Goldberg asks, did polyploids evolve naturally in plants, or were they hybridized in by people?
Doug, why don't you take that one?
That's a great question. So these polyploidy events that we're talking about are all, natural events, and they have occurred over and over. You might want to think about it as a rare event. It is, in a sense, kind of a mutation where, after a hybrid forms, something goes wrong in mitosis or meiosis, it happens, and then you end up with twice as many chromosomes. A lot of these events fail, but they're often successful. And you can imagine why, because you end up with 2 copies of everything. So it's better to have, maybe 2 pairs of shoes than one. You have one for inside, one for outside. And genes are the same way. You have an extra copy of a gene, you can use it for other things. But to your second point, do humans want to take advantage or employ polyploidy? Yes, we do. It's an incredibly important tool in agriculture. All of our crops are natural polyploids that formed, as I said, many, usually millions of years ago, sometimes 10, 100,000 years ago. But in plant breeding, there's a great interest in using that to take some things that are diploid and make them polyploid, because you may get better fruit production, you maybe get larger plants. So that is a great question. And yes to both things. It occurs naturally, and humans are trying to use it for economic importance.
Thanks. And thanks for that question. It's a great question.
Bonnie Basham, who is the president of the Florida Native Plant Society, I thought it was a question, but it's actually a comment. She says that apple skins /pulp/seeds can be boiled to make a natural pectin. Use it to help jelly and jam set up. And she says, then use the liquid and add to jams and jellies that have been made from low pectin fruits. Rombauer in Becker's Joy of Cooking explains the process quite well. And we're in Florida, which is the South, so in my opinion, everyone should have a joy of cooking at their house.
Yeah. And we do. Thanks. That's great information.
So I had a friend here in Osceola County. She runs the Osceola Gardener YouTube channel. She has many beautyberry plants at her house, and so she failed with the pectin. It did not set, and so she was going to throw it away, and I used the syrup, the beautyberry syrup, that resulted to make kombucha in secondary fermentation.
Oh, very
yeah. Someone in our lab actually did that. I don't know how it turned out though. She never got back to me. But I had one set, not the last batch I didn't set, and I just added more sugar and more pectin, and it set. So it makes sense.
That could be another mint minute. Another use.
Another use. I would be happy to join in on that mint minute demo a secondary fermentation on kombucha.
That would be great.
All right. Well, we're pretty light on questions in the chat. If anyone has questions afterwards, they can direct them to me. I think everybody at this point has my phone number and email address, communications@fnps.org, and I'll direct the question to the right person afterwards. So thank you everyone for joining us for this Lunch and Learn, and I'll see you guys next week.
Great. Thanks.
Thanks.
Bye, everyone.
Bye.